Okay, well, I'm very happy to introduce our very own Maggie Wagner. So for those of you, you all know Maggie, but you might not know that she did her undergraduate at Michigan in plant biology. And while she was there, she played hockey, goalkeeper. And I don't know how many times people scored, but I think she's. And then she did her PhD at Duke in bridging quantitative genetics with microbiology, which is the direction of her research. From there, she did a USDA postdoc at NC State, and then we were lucky to have Maggie here. So, Maggie.
So not a ton of quantitative genetics in this talk. This project's actually a bit of an outlier for that reason within the scope of what I do in my lab, but I haven't talked much about this, so I'm eager and a little nervous to tell you guys about it. This is my attempt at understanding this wonderful precipitation gradient that we have here in Kansas, that many of us have worked on in some fashion at some point. So my approach to my interest in this comes from my desire to sort of be able to integrate from these big ecosystem regional level patterns all the way down to the molecular level in plants. That's the type of thing that gets me really excited. So this is a talk about plant-associated microbiomes, so the communities of microbes that live in close proximity or even inside plants. And these microbes can perform a lot of functions that really matter for the plant. Things like contributing to the cycling of plant hormones, having big influences on plant phenotype in that way. General growth promotion, providing important nutrients like nitrogen, interacting with pathogens to protect the plant, things like that. So they can have very strong positive effects on the plant. They also do a lot of things that can have negative impacts on the plant, sometimes involving very similar mechanisms as the positive impacts that they have. And so because of their potential huge impacts on plant health, there's a ton of research interest in trying to understand these communities, how they work, and being able to predict what a given microbial community is going to do. Because there's hundreds, maybe thousands of species within a given soil microbial community, all with different suites of genes performing different functions. So how do you add up something so complex and understand what sort of function you're going to get out of it as it relates to a plant? And so that's sort of the motivation behind this bit of research that I'm going to talk about today, is trying to understand what are some of the processes and mechanisms that are going to lead from a microbial community to a plant phenotype. And so we can think about possibilities in terms of sort of the more proximate causes. Like if you give a certain microbial community to a plant, properties of that microbial community that might matter for the plant could have things to do with like the overall diversity of that microbial community, or which particular species make it up. What genes do they have? How are they expressing those genes? What is the environmental context? Are there lots of resources available? Are there not very many? And then on the plant side, there's genetic variation within a given plant species for this. Can they even perceive certain microbes over others? How do the plants express their genes? How do the plants influence which microbes can colonize them? And so forth. But there's also a lot more ultimate sort of explanations for a microbial community function that we can speculate about. Way higher level processes, like how did those microbes get to be where that particular plant is in the first place? And what does that tell us about that community? What has been going on in the evolutionary history of those microbes? What traits have been under selection? What have the patterns of gene flow among microbes looked like? And so forth. And then on the plant side, again, how has the plant in that particular location been evolving? What stresses has it had to adapt to? And a very interesting piece of this is, does it matter if the plants and the microbes have been evolving together for a long time, or at least evolving in the same location, and perhaps experiencing the same sorts of environmental challenges. So the goal of this project was to sort of explicitly link some of these ultimate causes of microbiome function to the more proximate causes that we can observe within an individual plant. So I asked this question: if we know something about the conditions under which a given microbial community has evolved, does that help us predict how they're going to interact, even on the physiological and molecular levels with an individual plant? So I tackled this question in the context of water limitation, as the environmental context, a common stressor that it has a big impact on both plants and microbes. And so in plants, a lot of us are familiar with the various types of adaptation that a plant can have to chronic low water availability, things like, think of a cactus, succulent leaves, the big root systems, different types of photosynthesis that conserve water, and so forth. There's also phenotypic plasticity, where plants can adjust their phenotypes in response to a sudden drought. And so things like, and this can involve some traits that affect microbes, such as increasing the amount of root exudation into the soil, increasing the thickness of the wax coating on the leaves, which forms an additional barrier to microbial colonization. Same with stomatal closure and so forth. And then the microbes have something parallel going on, right? Where a lot of taxa, like the actinobacteria, this is a streptomyces photo here. They have evolved adaptations to water stress, like thicker cell walls, the ability to go dormant. And they also have plastic responses as well to drought, where they stop consuming as much carbon and stop trying to acquire as many resources. And there's a huge pressure for them to accumulate solutes in their bodies at times of drought. But then as soon as the water comes back, as there a re-wetting event, it's very dangerous for them to have all those solutes in their bodies. And so there's high evolutionary pressure to be able to expel those quickly to maintain the proper turgor. So my point being that we have kind of repeated ecological shifts in community composition, favoring microbes that are adapted to deal well with low water. Also, natural selection happening within microbial lineages. And together, these things over time can build up to create what is often referred to as a soil legacy effect. Okay, so it's a bit of a nebulous concept, but it's this property of a soil microbial community that reflects the past climate that the community has been exposed to. And there's been a ton of research on soil legacy effects, soil microbial legacy effects, including in response to precipitation variation. And so there's plenty of examples of this happening. That they can have big implications for carbon cycling of other nutrients, many other ecosystem properties, but also plant growth. But one thing, despite this long history of research, one thing that is still a bit understudied in this field is looking at how did these soil legacy effects actually work? How do you get from this long-term climate variation to a change in the plant's phenotype that are causally linked? So that was the motivation behind this experiment that I'm going to talk about today. So this is just a high-level overview of what we did. We sampled soils from across a precipitation gradient, which I might refer to as the legacy phase. We took them into the lab, those soils, and we put them through a 5-month-long conditioning phase where we applied either plenty of water or an acute drought condition, and they either were in the presence of a plant host or without a plant host. And then finally, we measured their function using plants. So we actually inoculated them back onto plants to measure, to understand what were the implications of the legacy and the conditioning processes for plant phenotype. And so some of the readouts I'll talk about are the from the original soils, we looked at soil texture and mineral nutrient content, and we measured the metagenomes. During the conditioning phase, we also looked at the gene expression patterns in these microbes, as well as changes in the metagenome due to this conditioning process. And then in the test phase, we focused a lot on host phenotype and host gene expression. So I want to acknowledge the folks who were the core of this team that did this research. Dr. Nicole Gannan is a former postdoc here at KU. She's now working at the Penn State Microbiome Center. And then Gabriel, Valeria, and David are all researchers at the University of Nottingham in the UK. So this was an international collaboration to try to answer this challenging question. And I also want to give a shout out to Kansas NSF EPSCoR and everyone who is involved in the MAPS project from past years. I think you'll see why a lot of the MAPS work has inspired and informed a lot of the approaches we took. So trying to answer, do plants and microbes adapt together or separately to a shared stressor? First, we wanted to know, how do these soil microbiomes respond to water stress over the long term? Secondly, do these soil microbiomes react differently to an acute drought, depending on whether or not they're near a plant? And thirdly, how do those short-term and long-term responses to drought sort of feed forward to affect plant phenotype? I'm going to talk about this first question first, and then I'm going to kind of skip over the second one and then get into the plant phenotype. So many of us are familiar with this precipitation gradient in the central North America. From east to west, you have a very steep precipitation gradient with decreasing precipitation toward the west. So what we did was we visited these 6 sites and we collected soil samples from each of them. And these were prairie remnants that were part of the MAPS project. And so that's an important element of this is, as far as we know, these soils have never been irrigated or plowed. And so Tribune and Smoky Valley Ranch were our westernmost sites. And then we had Hayes and Kanza Prairie and Clinton Wildlife Refuge here in town and Welda Prairie a bit to the south. And from each of these sites, we took 6 sort of evenly spaced soil samples, and these were just our experimental replicates. We brought them back to the lab. So these were the sort of emergent molecular properties of these soils that we were interested in. So we extracted DNA. That tells us, first of all, what organisms are there, and second of all, what microbial genes are there. What's the functional potential within that group of organisms? And we also extracted RNA for some of our experiments, which tells us a lot more about which organisms are active out of all the ones present there, and out of all those genes in the metagenome, which of them are actually being transcribed and expressed. So we'll start with asking about what organisms are there, with the major caveat that in our data set, about 97% of our sequence reads were bacterial in origin. We got fungi, archaea, even some protists, and they'll make a brief appearance, but for purposes of this talk, I'm mainly going to be focusing on the bacteria for this reason. So our first goal was to identify the bacterial markers of sort of chronic water limitation, meaning low precipitation. And so Valeria used our metagenome data to sort of quantify at the broad level the changes in community composition as you go from dry to wet. And we saw sort of the typical expected pattern where you have more actinobacteria, now called actinomycetota, in the drier areas because they're commonly associated with adaptation to water. And that proportion decreases as you get to wetter environments. And that corresponds to an increase in the phylum formerly known as Proteobacteria. But we realized immediately there's a lot more than precipitation that is defining this gradient from west to east. So we wanted to do our best to control for some of the factors we know to be confounding. So Valeria took the approach of measuring texture, getting a measure of texture as well as mineral content using X-ray CT and ionomics with ICP-MS. And indeed, she found that there's a more or less a gradient of soil porosity. This is just in the first 5 centimeters of the soil profile. But you can see as you go from west to east that there is an increase in the soil porosity. So this is definitely correlated with precipitation. We need to control for it. The ionome was sort of the same deal. So here the different columns along the x-axis are all the different elements that she measured. And then the heights and directions of the bars just show the correlation coefficient with precipitation. So you can see the majority of the elements in the soil were either positively or negatively correlated with precipitation, sometimes very strongly so. And so her approach to control for these issues was to use a multiple regression framework to include those, control for them in the same model where she was trying to identify individual microbes that were correlated with precipitation. And so here on the x-axis, I have different microbial species. They're just identified with their ID numbers from NCBI. But again, showing the correlation with precipitation after controlling for those confounding factors of porosity and ionome content. So we have a bunch of organisms that were fairly strongly negatively correlated with precipitation, as well as a whole lot that were positively correlated with precipitation. So this was our first indication of sort of being able to narrow in on a robust and repeatable set of organisms that are good indicators of precipitation level. And so Valeria took the additional step of asking whether these results are just something specific to these Kansas soils, or is this a more general thing that that might take place elsewhere? So she went to the island she calls home, which is Santiago in Cape Verde. So this is off the west coast of Africa. It might be the first time that Kansas and Cape Verde have been compared in any context. I'm not sure. If anyone has counter examples to that, please let me know. I'd be very interested. But anyway, so she basically repeated this entire study on Cape Verde. So there are a couple properties of Cape Verde that make this an interesting contrast to Kansas. So first of all, the total range of precipitation variation is a lot smaller than in Kansas. There's still a nice linear sort of gradient that she can work with, but it's within a much smaller geographic area, which helps to minimize some of those confounding effects. And also, unlike in Kansas, the gradient's not totally linear, which is a nice feature. You can see that well, you can't really see on this map, but they're volcanic islands, and so the center is higher, and that's where most of the rainfall catches. It mostly is coming from condensated ocean mist. And so it catches on the islands in the center where it's wetter. By the time you get down to the coast in any direction, it's drier. Okay, so we don't have this sort of spatial autocorrelation problem as well. So she repeated that, and what she found was that out of the 14 bacterial taxa that we found most strongly correlated with precipitation in Kansas, 8 of those were validated in the Cape Verde precipitation gradient. And of the ones she identified in Cape Verde, 11 out of 19 were validated in Kansas as well. And then there were 3 particular species that popped up independently in both precipitation gradients. So we think those ones are the most reliable markers of precipitation. So from this, we would conclude that there's some elements of precipitation gradients that are effects on soil microbial communities that are congruent even in very far-flung locations. So we used our metagenome data to next look not just at which organisms are there, but what genes do they actually have, okay? And so this is where David Gopalchin took over the work. And I'm not going to dwell too much on this because the interpretation of it is not super clear, but suffice it to say that we found um, a relatively large number of gene categories, and not to mention individual genes that were strongly enriched along this precipitation gradient after controlling for everything else. And so just some very high level general patterns. There was a lot of stress related functions like DNA repair that were more common in the dry legacy soils. And in the wet legacy soils, it seemed to be, we noticed a lot more functions popping up that were related to cellular communication, perhaps between organisms, or perhaps even with a plant host. So we can kind of make up a story where that makes sense, because we expect there to be a lot more plant neighbors present when there's high precipitation. But even cooler, in my opinion, David went farther, and in addition to just comparing the total genes present in this soil versus the total genes present in that soil, he actually sort of narrowed in on individual microbial species, and actually was able to find strain level variation across the precipitation gradient. So this is notable because now we're talking about not just about comparing communities to each other and their species composition where the species have different genes. We're talking about within a single microbial species or lineage, how much genetic variation is there? And that's important because that's the material you need for actual genetic evolution and adaptation. So he focused on 33 species that had sufficient sequencing depth and were abundant enough and prevalent enough in our data set to be worth kind of digging into. And so what I'm going to show is a Manhattan plot where along the x-axis, these are bacterial species. Usually when you see these, they're representing chromosomes within a single organism, but here we have one species at a time. And each point represents a single nucleotide polymorphism, and its height along the y-axis tells you whether it was significantly associated with precipitation, okay? And so here's what the full data set looks like. All these red points highlighted above the false discovery rate indicate single nucleotide polymorphisms that we have evidence to show that these are significantly associated with precipitation. So these produce basically candidate genes, bacterial candidate genes for adaptation to these precipitation gradients. And these were from 6 different species, including 2 Bradyrhizobium. Okay, so leaving the legacy phase, that was all about our observations from these naturally occurring soils from across a precipitation gradient and what might be going on there. We're moving on now to the world of experimentation. So at this point, we brought all these soils into the lab. We divided them up into pots, microcosms, I guess, and half of them got a plant host. The plant host was Trypsicum dactyloides, which we'll meet again later. And they got either plenty of water or very little water for 5 months. Okay, and so we compared afterwards the metagenome content after the conditioning process, as well as before and after conditioning metatranscriptomes in the lab. Okay, so this is the experimental design I just described. Just a reminder, these were done with 6 different soils, 4 treatments in a factorial design, and 5 replicates. This is sort of what the experiment looked like. I think this was shortly after a watering, but each of these pots will have had a different soil microbial community in it. I'm not going to dwell too much on this part of the experiment because, well, we're still working through the data, and we made decisions to focus more on the legacy effects for now, but I'll just kind of give a broad overview of our early observations. So every taxonomic group that we detected in these communities had a significant response to the drought treatment during the conditioning phase. So you can see each of these points represents a bacterial community points that are closer together are more similar in composition than ones that are farther apart. And the green ones are the baseline, so those are the communities we measured in the field soils. And then the drought in brown, the points in brown, are the experimental communities after experiencing that 5-month drought, and the blue ones are after are the control group. And so you can see very clear differentiation between those two treatments in the bacterial community. In bacteria, it explained about 4.3% of the total variation. All of the other taxonomic groups had a weaker effect, but it was still detectable. So fungi, we saw it about 3% of the variation. Archaea, we saw the same thing. And I'm gonna just gloss over this, but we also saw in all of the groups a clear differentiation of the communities depending on whether or not there was a plant growing in their pot. So bacteria, fungi, archaea all showed this pattern. So did the protists. But like I said, this is very much a work in progress. We have some very cool questions that we're really looking forward to digging into, but we haven't quite had the time yet. So we want to know, are those shifts in the community due to that acute drought? Do those share properties with the patterns we saw along the precipitation gradients? Or is it kind of different processes driving that? And of course, we're interested in the interaction between host presence and drought treatment. There's evidence out there that some of the traits that are selected for in microbes to adapt to low water conditions include the ability to degrade complex polysaccharides, such as the type that plants like to dump into the soil. And so there's reason to believe that those things might affect each other. But for purposes of this talk, this is the main takeaway I want you to get from the conditioning phase, which is that even after that 5-month drought period, with or without a host, the signatures of the precipitation legacy were still very obvious, even after the conditioning phase. So these legacy signatures were pretty robust. And so this is showing the post-conditioning metagenome with now the dry legacy versus wet legacy soils. Same thing with allelic diversity. So same as the microbial GWAS, the patterns of within lineage genetic diversity were still totally distinguishable. And the strongest of all was the metatranscriptome, still very, very strongly differentiated between dry legacy and wet legacy communities. So to sum up what we learned from these soil bacteria, my favorite is that we detected clear evidence of both ecological processes and evolutionary processes within microbial lineages contributing to these precipitation legacy signatures. These signatures of precipitation legacy are robust, especially at the level of bacterial gene expression. At least some of those signatures of precipitation legacy were congruent in Cape Verde and Kansas, which you would expect to have nothing in common. So that kind of increases our confidence that this could be a more generalizable pattern. So having addressed very briefly those two questions, I want to move on to how these soil microbiomes responses to precipitation and to water stress in general, how do they feed forward to impact the plant phenotype? So I'm going to present data from 2 different plant species. The one on the left is eastern gamagrass, Tripsicum dactyloides. It's a very big perennial bunch grass that's native to a lot of eastern North America. I think the westernmost accession is actually from western Kansas, and so it's like in the Lake Scott area. So we think it has more or less been evolving within the same region that that these soils were taken from. And then as a contrast, we have maize, which many of us are familiar with, very important crop. It's also quite closely related to eastern gamagrass. It's not the progenitor from which maize was domesticated, but it is the closest evolutionary relative of maize that is native to temperate North America. Okay, so the progenitor of maize is tropical, did not evolve here. Eastern gamagrass is the next closest thing that fits that bill. And you can kind of see a bit of resemblance in the flowers, which are actually really beautiful, in my opinion. Okay, so the conditioning phase plants were Eastern gamagrass. And so if we wanted to understand the effects of these communities on plant phenotype, well, why didn't we just measure the plants during the conditioning phase? Well, we did, and we saw some sort of intriguing patterns, like as you go from west to east, the shoot biomass of those plants under the drought condition drops fairly linearly. But the problem with this is that, as we discussed earlier, there's a lot of confounding factors in these soils that made us a little kind of doubt whether this was only just due to a precipitation legacy, or it's probably, who knows, it's the particular mineral compositions and all that other stuff as well. So what we did to get around this was we conducted a third phase of this experiment where we took the outputs of the conditioning phase, so the soils after their 5 months of conditioning with or without a host and with or without water. And we isolated the microbial fraction through centrifuging and other methods and inoculated a brand new set of plants where we controlled for the soil medium. It was like an artificial, like a calcined clay medium. So very homogeneous among all the pots. And so by doing that, we were able to remove any association between the microbial communities and any sort of physical or chemical properties of the soil that could confound our results. And for this, well, we have a lot of the phenotype data from all 6 of the soils, but we opted to focus on the 2 extremes because, first of all, we were limited in how much of some of the molecular data we were able to collect. And secondly, there were some properties of the 2 soils near the center of the gradient where things started to get a little fuzzy and some signatures of the precipitation gradient extended easily through that zone while others didn't. And so to give ourselves the clearest point of comparison, we decided to focus on the 2 soils from either end. So after the conditioning phase, we took those and we inoculated those into pots, and we planted a plant seed. That's what that is, obviously. And we let them grow. So this is so the word phytometer, it's exactly what it looks like, using a plant to measure something. So we're using the plant's phenotype to measure the function of the community. And so here are some photos from the experiment itself. So you can see a lot of variation in plant size. We only grew the plants for a couple weeks because we needed a lot of them and we had small pots and we didn't want them to get root bound. You can see from the shapes of their root systems, they were a little constrained, but were still growing when we collected them. So they were not actually root bound. So some of the traits, in addition to many sort of standard phenotypes that we measured, we also looked at gene expression within the crown roots of both species. We looked at nutrient acquisition using ICP-MS in the leaves, photosynthesis, root system architecture, and root anatomy as well. And root anatomy was also focused on the crown roots. And I forgot to put this on here, but we also did amplicon sequencing of the bacterial microbiomes, which I'll come back to in the end. Okay, so we had the 4 soil inocula, 2 from dry regions and 2 from wet regions, factorially crossed with a drought treatment or a control treatment, and then replicated in both plant species. We confirmed that our drought treatment was sufficient to really knock down plant performance, which gave us confidence that this was a successful manipulation. And so we had a lot of measurements. And so we decided to really focus in on drought tolerance. And we used an S-index approach, which has been used elsewhere in the literature. Essentially, what we did is we standardized all the plant phenotype data of the droughted plants by the mean of the well-watered plants within the same treatment. Okay, so it's a ratio. And so it'll look something like this. Our scaled trait values, or S index. If it has an S index of 1, it means that the drought-treated plants were phenotypically identical to the well-watered plants. But if it's less than 1, it means that so like we might observe an S index of 1 given one microbial community. We might observe a different S index in a different microbial community. So what that means is that in the presence of this microbial community, the droughted plants had only 85% the trait value of the control plants. So that's how I've presented a lot of these trait data. And so we measured so many traits. Before we did anything else, we looked at, which ones were highly correlated with each other. Nicole used a random forest approach to identify the most important functional traits for explaining drought tolerance. And so we only ended up doing this analysis for a relatively small number of traits. But for eastern gamagrass, 7 out of 9 of those, we found a significant effect of the microbial precipitation legacy on the S index of those traits. So transpiration, for example, in the presence of either of the two wet legacy communities, transpiration was down to like 27% or so of the controls, where in the presence of a dry legacy inoculum, it was closer to like 37%. Same with intrinsic water use efficiency, increased from like 45% to about 65% in the presence of a dry legacy community. Chlorophyll content, we actually saw that in the presence of the dry legacy inocula, chlorophyll content was actually even higher by quite a bit, by like 20% than the control plants, which was interesting. And we saw similar, we saw patterns as well in various root traits. One of the things that confused us at first was we found that the dry legacy communities cause the plants to have relatively smaller root systems compared to the wet legacy. However, after reading up on it, and we were especially confused about this metaxylem area, because the metaxylem are these little, like in this cartoon, these blue dots. That's, how the plant draws up water. Seems like it would be important for drought, but it turns out there's precedent in the literature for smaller metaxylem being a trait associated with drought tolerance, because it reduces the risk of cavitation, where you have air bubbles forming in there, which can be deadly for a plant. And so that's what we think might have been going on there. And in total, about 5% of the phenotypic variation in eastern gamagrass could be attributed just to the precipitation legacy of the soil microbiota. And at the gene expression level, we found a handful of genes that were upregulated or downregulated in response just to the inoculum. But way more interesting than that, it was the interaction between the drought treatment and the inoculum. So what I'm showing here, individual dots will represent individual genes. And if they're to the right of this line, positive values, it means their transcription was increased in response to drought. To the left, the transcription was decreased in response to drought. So for example, in the presence of a wet legacy inoculum, this particular gene got downregulated in response to drought, whereas in the other inoculum, it was upregulated, okay? Or you might see a pattern like in one inoculum, it had no change, right? It's overlapping 0. But in another, it did go one way or another. And another pattern that we sometimes saw was that they were always changing in the same direction in response to drought, but the magnitude of that effect would change depending on the inoculum. So here are the actual data. I'm only showing the stronger the stronger fold changes. So in total, there was almost 200 genes that showed a significant interaction here. So this first group was largely characterized by genes that had no response to drought in the presence of the dry legacy inoculum, but they decreased their expression when the plants were grown with the wet legacy inoculum. And these included some interesting ones that were linked to abiotic stress tolerance. Well, I don't want to read too much into it. But I think the overall more interesting pattern here is that the main groups of genes that popped up in this analysis showed this pattern, which you can see here with all these, the brown points overlapping 0 and the blue points on either side of it. What this is telling us is that the most common pattern we saw is that inoculation with these microbes from a dry climate tended to sort of tamp down the transcriptional response of eastern gamma grass to drought. And so I wanted to try to tie in this transcription data more explicitly to the phenotype data to try to connect some dots here. And so I did a mediation analysis, which is a flavor of path analysis or structural equation modeling. And so this was essentially our model of how things worked, where we have an acute drought, which would directly affect the plant trait through any of the, we detected like 8,000 genes that changed in response to drought. That's not surprising. But then we had this one set of the genes that were sensitive to the microbial legacy. So that's, all of these genes. So that set of genes versus all the other genes, we were able to tease apart, the relative importance of that particular set of genes on the plant phenotype. Okay, so that's the effect of those legacy sensitive genes will be shown in blue, and the direct effects of drought on the plant trait in yellow. Oh, and I quantified these legacy sensitive genes. I summarized them just using multidimensional scaling, and I picked the top 2 axes of that ordination to represent the overall expression of that set of genes. And so we did this for each of the 9 focal traits that we were looking at. Only 2 of them showed a significant evidence of mediation by this set of genes that we know to be responsive to the microbial legacy. But those 2 traits were good ones. We saw an effect on intrinsic water use efficiency and also transpiration. So what these yellow bars tell you is that the direct effect of acute drought had a very strong effect on negative effect on both of these traits. The blue bars show you what was the contribution of the microbiota sensitive genes to that change. And so you can see that between 9 and 18% of the variation or of that change could be attributed to the influence of those genes, depending on which trait you're looking at and which component of the that set of genes you're looking at. And interestingly, we had this one MDS2, so that component of gene expression, you can see it had an influence in the opposite direction of the effect of acute drought, which tells us that it was having some sort of I don't want to say protective, but it was sort of slowing down or interfering with the effect of the acute drought. And so I looked more closely into that MDS2 component of the transcription data, and I looked at which genes were strong contributors to that particular axis of variation. And 4 out of the 8 top genes contributing to MDS2 were orthologs of this maize gene, which is a nicotianamine synthase. More evidence that these are actually important in plant responses to microbiota precipitation legacy is that all of 4 of those genes were upregulated, sometimes very strongly, like 12-fold in response to drought. So plant experiences drought, it ramps up its production of these, or transcription of these genes, but that only happened in the presence of the dry legacy biota. It had no response to drought in the presence of the wet legacy biota. Furthermore, two of those genes were in the strongest negative contributions to MDS1, meaning that they would have been pulling this blue bar up, decreasing that transcriptional pathway to the acute drought response. And so a bit more about this gene. I've seen enough now to say this is a viable candidate gene that would link the soil microbiota precipitation legacy to the plant drought response in eastern gamma grass. So this is an enzyme that produces nicotianamine, as its name suggests. Nicotianamine is mainly involved in metal chelation and iron acquisition in the soil, but there's, and as far as I could tell, it had not been really specifically linked to microbes, but although its expression is known to be induced by salicylic acid, but so are a lot of things. Salicylic acid is a important plant hormone and is known to be one of the major regulators of root microbiomes. I also found two studies that had linked this gene to tolerance of acute drought, one in rice and one in perennial ryegrass. Both of these were transgenic studies, overexpression of the gene, but it just struck me how similar their results were to what we had observed in the eastern gamma grass inoculated with the dry legacy community, specifically the ability to maintain photosynthetic activity in the face of acute drought. And in the ryegrass, they specifically noted that plants overexpressing this gene even during severe drought had more chlorophyll than the control well-watered plants, which is exactly what we saw in Tripsicum. So it's certainly intriguing, but obviously this needs a lot more work to sort of confirm it. Okay, so what about maize? I kind of stopped talking about maize. Well, in a nutshell, what we saw is that the effects of inoculum precipitation legacy on maize were just across the board weaker than the effects on eastern gamma grass. So we still detected an effect, but it only explained about 2% of the total phenotypic variance. The particular traits that we saw an effect for were not particularly interesting. There's some metal acquisition seemed to be affected. The maximum number of roots, we saw plants with the dry legacy community tended to have more roots than the wet legacy community. We found fewer, but still some genes that were differentially expressed. So there was 23 genes upregulated in response to the dry legacy community. We did note, this is kind of anecdotal, because we could, unfortunately, for various reasons, couldn't directly compare gene expression profiles between the species. But we did notice a lot of defense-related genes popping up in the maize data set where they did not pop up in the Tripsicum data set. And the same was true for the interaction term, the drought responsive genes. We had a bunch related to pathogen defense, but also several related to symbiotic fungi, which was kind of interesting. And that sort of suggests that both enemies and friends are parts of this precipitation legacy process. But this observation that Tripsicum benefited more strongly from these dry legacy communities, this is consistent with the expectation that has been put forward that these positive effects should be stronger in pairs of interactors that have co-evolved or come from the same place at least. And just a brief kind of side story, which I think is neat, is there were some drought responsive genes in both species that could only be detected after accounting for the soil biota precipitation legacy. So like I said, there was like 8,000 genes changed in response to drought stress on average, but there was a bunch that only showed the interaction term. And there were some good ones in there, like this aquaporin, which regulates water transport in and out of the vacuole, which had pretty strong changes in response to drought in either microbiota. But if you were to just average across the microbiota, you do not detect it at all. And so what this tells us is that if you want a full kind of accounting of what's going on molecularly in a plant, it would behoove you to know a little bit about the microbiome. And I'll end, I think, on kind of just a description of what was going on in these root communities. So like, trying to link now, these biota and legacies, how does that translate into the plant's phenotype and transcription? So this is just a 16S amplicon sequencing from the crown roots. What we saw is that in Eastern gamma grass showed a very strong homogenizing influence over the microbiome, such that there was almost total overlap in the root bacterial community, regardless of which inoculum it had received. Whereas in maize, that signature of inoculum legacy was still very obvious. And so that kind of belies the phenotype patterns we saw, where Eastern gamma grass had the strongest response, and yet it's within the root, there seems to be very little distinction between the two inocula. So we're not really sure why that is, but it's pretty interesting. And then just generally speaking, what we saw was that in response to the drought treatment, the bacterial community structure in maize roots was very, very sensitive to the drought treatment. So that to see that, you look for the triangles, which are the maize. So the blue triangles down here are the well-watered. The green triangles up here are the droughted maize plants. And then these other two ellipses in here are that overlap almost completely with each other are the gamma grass microbiomes in droughted or well-watered conditions. So you can see the gamma grass microbiome was super stable, didn't really care about having enough water. And we saw that as well at the level of like alpha diversity and even beta dispersion. So that's like how much like noise essentially is within these microbial communities. There was no effect in Eastern gamma grass, but in maize, the application of drought really dropped that alpha diversity and really blew up that the beta dispersion. Okay. So what do we learn from our phytometers? The precipitation legacy explains at least some of the emergent functions of a soil microbial community for plant performance, particularly in the context of a subsequent acute drought. And so we can see these signatures of climate legacy at the molecular level in these plants' roots. And these effects are not necessarily transferable across species, maybe, but not necessarily due to some coevolutionary history. But that's relevant because, a beneficial soil will not be beneficial for everything. There's a lot of work to do still on figuring out some of those causal links, especially from the microbiome composition in the roots to plant gene expression. It's not clear why we observed what we did. So I'll thank again, especially Nicole, Gabriel, Valeria, and David for their amazing work on this. Many other people in my lab also contributed to this. And I just want to end with a plug for 2 upcoming talks on our favorite species, Eastern gamma grass. So Joel Swift will be talking a month from now, and Jada will be talking 2-3 months from now on various other aspects of Eastern gamma grass biology and interactions with microbes. So something to look forward to. I'm happy to take any questions. So Jim's question was about the our observation that there was very little difference in the root community composition, and yet we saw big phenotypic effects. And so what might be causing that? Rare organisms is a possibility. Other possibilities that I have thought about include, well, first of all, these were from inside of the root. Perhaps the relevant processes are happening outside of the root on the rhizoplane or in the rhizosphere. Another possibility is that It involves microbial behavior. You know, amplicon sequencing can only tell you who is there. You can't tell, you know, which ones are active. If we had been able to measure gene expression, for example, we could have observed something totally different. And so unfortunately, it's very difficult or really basically infeasible to do that inside of a host because of all the interference from host nucleic acids. And so we can't really get that data. But that would be probably one of the first things that I follow up on. Sharon. Sharon's question was about the water use efficiency results and what are the possible mechanisms and might they involve the leaf microbiome as well? Yes, that's totally a possibility. To be honest, this is something I have a lot fewer hypotheses about. We are starting to look into things like stomatal behavior and stomatal density above ground traits that have to do with water usage. But we didn't really in this study. And so I'm very hesitant to kind of put that forward. I think the earliest data we have on this from our lab is we see a ton of genetic variation for stomatal density and so forth, but not very much effect of the microbes on it. And as far as other above ground functions of the above ground microbiome that could be contributing, I don't have very strong intuition about how exactly that would work. Do you have particular like functions of those epiphytes in mind? Pure speculation. Yeah. Yeah. Nitrogen cycling processes did come up in our metagenome comparison of the dry versus wet legacy soils early in the talk. Yeah, that's a good suggestion. Yes, Ben.
Thanks for a great talk, Maggie.
Thank you.
So I have a question. I know you have more work to do on the transcriptome that you found in the second section of your stuff. It looked like, first of all, I would expect the transcriptome to have the least kind of carryover of the soil legacy, right? Because you'd impose stuff in transcriptomes. We generally think are changing faster than genes, right? It seemed like you still had clusters in that transcriptome. Are those, can we assume that those are the three legacies, the control, the wetter, and the drought? Is that what those are?
Yes, exactly. Yeah, so Ben's question was in the metatranscriptome data after the conditioning phase, there appeared to be clusters in the gene expression, and did those clusters correspond to the 6 original soils? The answer is yes, basically. And that's kind of expected, right? With the different, we know that the compositions of those communities were different to begin with, and so the suite of genes being expressed will also be different. What we didn't necessarily expect is that they would be more different than the composition of the communities. To me, that says something about, you know, part of what distinguishes these communities is actually in their response to environmental stimuli.
That same result, I mean, I was looking for evidence, any evidence that your training period mattered, and I was imagining those clusters had to do with that, but so what did the training all of that time, plant, no plant, high, low, water, did it matter at all?
It did matter. Yeah, we found
Yeah, that's what you just said is that it was the 6 legacies, but I was thinking it was the clusters were.
Oh,
yeah.
oh, okay. I completely glossed over the conditioning phase because, okay, wrangling this experiment was very challenging, and we decided to like, okay, let's just focus on the legacy for now. So to be honest, we haven't even really looked heavily into this yet.
So all these effects are just consistent across all of those 4.
Yes, exactly. That's the takeaway. It's every result I showed in the second half of this talk is the average across multiple soils per legacy and the average across these conditioning treatments, 4 different conditioning treatments, which we we do know enough to be able to say we saw very clear changes in the metagenome and metatranscriptome content of those communities because of the conditioning. And yet we were able to find these really consistent signatures of precipitation legacy in the plant. It's cool, right?
And I thought bacteria didn't matter.
Hmm.
Good try, thank Maggie again.
Thank you everyone for coming.